My name is Asia Smith. I attend Howard University. I'm a rising junior. I'm a biology major and a chemistry minor. My project this summer is focused on exploring how early life adversity affects long-term behaviors. And we do this by identifying a group of cells during the early life of mice. And then later on, we do behavioral testing to see how these cells are reactivated or intake as they undergo. The part of the project I'm most interested in is the fluorescent confocal microscopy and counting cells, because those are two areas I haven't really engaged with thus far in my research journey, and being able to see how the behavioral aspects of the mice works on a cellular level is very interesting to me. I'm most looking forward to being able to gather all the data together using the reward testing as well as the cell counts and the images to see how exactly early life adversity can appear on a cellular level and what is this mechanism that is going on and the further implication of the whole experience. I enjoy going to the gym every morning. It helped me establish a lot of discipline and learn how to really put effort into the things that I want to do. I also enjoy reading and spending time with my cohort members and doing fun things, just creating them. One highlight from the summer thus far has been going to Laguna Beach with my cohort mates for the first time. I'm from the East Coast, so I don't go to a lot of beaches, so it was a really fun experience. And it was my first time at a West Coast beach, so I was able to just enjoy the sun, read, and then just mingle and listen to music, so it was just a really good day.
My name's Mason Hardy. I'm a fourth-year graduate student in Dr. Tallie Z Baram's lab. I am working on a project that is investigating the impact of early life adversity on the hair ventricular nucleus of the thalamus. This is a region that we have recently identified as a potential encoder of some of the salient stressful aspects of ELA that we believe may be actually contributing to some of the long-term behavioral effects that result from this exposure to early life stress. So a lot of my project is actually building on some prior work from Cassie Koiker, a former graduate student in our lab. She's the one who really identified the paraventricular nucleus of the thalamus as a region that is preferentially and almost uniquely activated by early life adversity. And we are now manipulating those cells in adult animals to see if we can actually change their behavior and sort of rescue some of the behavioral changes that we see in ELA animals. We don't know what the activation of these cells in vivo or at the time that mice are being exposed to rewarding stimuli. So what Asia has been working on has been to look within the brain at the level of cellular activation within these cells that were also active during this early life period. It's been really great having Asia working in the lab. I think she's been phenomenal working in the lab. She's super quick to learn techniques. I'm learning a lot from her. I'm really enjoying the experience. When you first start as an undergrad, everything's really novel and new. Having somebody in the lab that you can sort of introduce to these techniques and they can experience it for the first time, it's really fun seeing that sort of excitement and that joy of like learning a new technique and how just fun and cool it really is for these people.
We would like to know how early life adversity, how things that happen to the brain when it's really plastic, when it's really growing and developing, how to change it.
Because if we know what happens, we then can figure out how to prevent the bad stuff and maybe to omit good stuff that happened to the brain.
And we know our children change so quickly, the brain changes so quickly.
So this is really the area that we're focusing on. And the kind of outcomes that we're looking at is things that are related to depression,
to loss of pleasure, to drugs taking.
So we bring in people from many, many parts of the world and many parts of the country
to really do that kind of work. We use really sophisticated methods. We have ways to light up the brain, to shine light through it, to manipulate parts of the brain, to get them to work, to get it to stop to work,
to understand this incredible computer that we have.
So that's really what we're doing.